Perfect. Great. Thanks everyone for joining. I think we can go ahead and get started. It's great to see so many of you here. And yeah, as you've been hearing us discuss, we're using the new webinar feature here. So, please, type in chat if there's any technical difficulties, if you have questions, if you just want to say hey. And I will go ahead and kick us off. Welcome. For those of you who don't know me, I'm Brooke Babineau from the KCNQ2 Cure Alliance. I am the scientific director, and I'm really glad you're joining us tonight to hear about a really exciting topic about therapeutic development in the KCNQ2 space. We know a lot of you have had questions and interest about therapeutic development and maybe have heard the letters ASO used and wonder what they exactly meant. And so we will start that discussion tonight, and it will be an ongoing one as there is a lot to take in about genetic medicines and how we're approaching them. So today we're going to be joined by members of Dr. Tim Yu's lab at Boston Children's Hospital. The Yu lab focuses on genetic medicine for rare diseases, including the development of individualized antisense oligonucleotides, which is what ASO stands for. So this team combines advanced genetic sequencing, bioinformatics, and translational research to better understand rare genetic disorders and explore possible therapeutic approaches. So our distinguished panelists tonight include Margaret Meserve, a genetic counselor in the U Lab, who will explain what ASOs are and how they work. Emma Sherrill, the research data manager, who will walk us through long-read sequencing and why it matters. And then Aubrey Susi Varon, the senior bioinformatics scientist, who will talk about how sequencing connects to ASO development and current KCNQ2 research efforts. Just a few housekeeping notes, which we might have touched on before, but as some new folks might have joined, please feel free to drop any questions in the chat at any point during the presentation. We can come back to them during the Q&A at the end, or for those questions that I can potentially answer during the conversation, I will do so. If you prefer to ask your question live at the end of the session, you can use the raise hand, and we will either unmute you from our end or invite you to unmute yourself. And we are recording today's session, actually, and Marissa can maybe just confirm that we have started the recording. We are recording today's session, and we'll share the recording afterward on our YouTube channel. So with that, we will start with Margaret, I believe, and then the team will carry on with their presentation. Thank you very much for being here, both panelists and audience. Go ahead.
Perfect. Yeah, thanks so much for having us. We're very excited to get the chance to talk to you all today and hopefully have some time to answer your questions. So to jump right in, as we mentioned, as Brooke mentioned, our agenda today is to give you a little bit of some basic information about what are ASOs, why are we talking about them, to understand kind of how long-read sequencing connects to the ASO development pipeline, and then to give you a little bit of an update and some information about the current status of some KCNQ2 ASO programs that we know about. So before we go into ASOs, I do think it's kind of helpful to set a little bit of a genetics background just to refresh ourselves and to make sure that we're all on the same page. So as you may know, and you've probably heard before, our DNA provides the instructions for how our bodies grow and develop. But DNA actually has to go through several steps before we have a product that actually can do all of the actions in our bodies to support our daily life. So first, our DNA gets transcribed into RNA, which is like a simpler copy, just the instructions that are needed at a single moment in the cell. And then those instructions are used to create proteins, which are what actually do the work, the stuff that our cells need. Next slide. A good analogy for this is actually a cookbook. Our DNA or our genome is like the full cookbook. You've got lots of different recipes, tons of pictures and stories. There's just a lot of information. RNA is like the recipe that you want to cook for dinner. It's the specific instructions that you need to make the dish that you'd like, and it tells you what to cut and what to mix together and all the steps and ingredients that you need. Finally, a protein is like the finished meal. If your cookbook has all of the right pages and you followed all the instructions, you have the dish that you were expecting to make at the right time and the right place. KCNQ2 related disorders occur when there's a genetic change in the DNA of a gene called KCNQ2. Makes sense? In our cookbook analogy, KCNQ2 provides the recipe to make a very specific and important protein for a potassium channel that's found in the brain. These potassium channels help maintain and regulate the electrical activity in our brains. And some genetic changes in the KCNQ2 gene are actually significant enough to make the recipe hard to read or even impossible to read. For example, we all have 2 copies of every gene, one from mom and one from dad. Some genetic changes cause a loss of function where no protein is made at all. Other changes are what we call dominant negative. You can see right here, where the altered protein also impacts the normal protein that's made by the healthy copy of the gene. I'm going to really quickly just make a note here that loss of function is used slightly differently in genetics than it is in neurology. So sometimes these vocabulary words get tossed around all the time. They're actually used slightly differently. Both of these variant types actually result in a loss of channel function. So your potassium channel isn't working the way that it's supposed to. But when we say loss of function in genetics, we typically mean that there's no protein being made at all. That's different than dominant negative. Okay, so now that we've established some of the vocab and we've talked about genetics, I'll get into what are ASOs. So we're here today to talk about a specific type of therapy that belongs to a larger group called genomically targeted therapies. These are drugs that are made to specifically address the impacts of a genetic variant in the body. There are several different types of genomically targeted therapies. The two that you might have heard quite frequently is gene therapies is one. Gene therapies modify a person's genetic material directly. So they work on the DNA. These are approaches like gene editing or gene replacement. They fall into that category. On the other side, ASOs work downstream of our DNA. So for example, ASOs typically target somebody's RNA. So it's still within this pathway, but they're not changing the DNA itself. They're not making permanent changes to a person's DNA, but rather helping modify the recipe. There are several different types of ASOs. The full name is antisense oligonucleotide, but it's a lot easier and shorter to say ASO. There are several different types that can be used to address genetic changes. Our lab primarily focuses on 2 different types, splice modulating and knockdown ASOs. Today, we'll be talking primarily about gene knockdown, or also known as gapmer ASOs, as this is the type that is most applicable to KCNQ2 related conditions. And for that reason, dominant negative variants are actually a particularly good target for knockdown ASOs. As you may know, as we talked about, you have 2 copies of every gene. And as we said, with dominant negative variant, the mutant protein that's created as a result of the variant actually impacts the functioning of the normal protein. So it's not working as well as it should. We can use a knockdown ASO to essentially flag the copy of RNA that has the genetic change in it and tell the body to get rid of it. And we say, this isn't helpful and it's actually hurting the other copy, so we're going to get rid of it entirely. This allows the normal protein to continue working as it should without interference. So when we discuss the potential of designing an ASO for a specific condition, like KCNQ2, we start with what we call a therapeutic hypothesis, which essentially means that we are looking for a way to use ASOs that we think will result in some sort of clinical benefit or reduction in symptoms or just something that would be helpful. For KCNQ2, we start by, again, saying that individuals without any genetic changes in KCNQ2 most likely do not have any symptoms of the condition. Their proteins are working at 100%. The ideal situation in drug discovery would be to design a therapy that would return the genetic change back to the state, right? To get back to 100% function of the protein. Unfortunately, ASOs are actually not able to do that for KCNQ2. So we look for other options. Strategies like gene replacement or gene editing are options that would get to 100% function, but those aren't quite ready for prime time yet when it comes to neurologic conditions. So variants in KCNQ2 actually cause two separate clinical presentations. Generally speaking, a loss of function variant or the loss of function form has been known to be associated with something called benign familial neonatal seizures. These individuals have about 50% function of KCNQ2 protein in their cells. Clinically, they typically will have seizures early on, but sometimes, those will eventually stop. And they tend to have a slightly more mild presentation compared to others. On the other hand, the dominant negative form is associated with neonatal epileptic encephalopathy, or a DEE. These, like we've talked about, have both copies are impacted just by the one genetic variant, and they have less than 50% KCNQ2 functioning in their bodies. This tends to result in a slightly more severe presentation with seizures that last longer into childhood. I'll mention quickly that this is an oversimplistic explanation. In KCNQ2, we've seen some cases that seem to blend these two different clinical pictures, or genetic variants that have been reported to cause both either or diagnoses. And so it's not exactly crystal clear one way or the other, but it's something that we're still trying to look into. But our general hypothesis is that an ASO could be used to knock down a dominant negative variant. This would result in roughly 50% KCNQ2 function for individuals who previously had less than that. So it would create a little bit more KCNQ2 function for those individuals, and would be a similar genetic scenario to the benign familial neonatal seizure symptoms. However, how this would actually look in the clinical trial is something that we don't quite know yet. And clinical trials are kind of how you answer that question. Just to define what a clinical trial is, these are experimental studies where a new therapy is given to humans, often for the first time. The goal of these studies is primarily to understand if a drug is safe. Further testing and long-term studies will often tell us if it's effective in managing symptoms as well, but usually the first primary outcome of a clinical trial is understanding safety. The study design and who might be eligible for a clinical trial is highly dependent on the therapy itself. For ASO studies, we've seen some cases where small groups of patients are enrolled, and others where the ASO is highly specific and might only work for one or a very small group of people. And so that all kind of depends on the therapy itself. Just for some more information specifically about ASO clinical trials, ASOs are typically given via lumbar puncture or spinal tap, as they're more familiarly known for some people. These are often given in an OR under sedation. Adults can often tolerate this kind of procedure at a bedside, but for kids, it tends to be in an OR for safety reasons. Dosing typically occurs every 2 to 3 months, with early phases kind of being more frequent. This is important to know because this type of therapy is given indefinitely. It's not a one and done, and the goal of the therapy is to continue to give it over time. And so that's just one thing to think about. Clinical trials for ASOs often will include additional study visits outside of the dosing visits for things like safety monitoring and other evaluations. This could include things like blood work or MRIs and EEGs, or other neurologic visits or tests. And costs associated with clinical trials are generally covered by the study sponsor. However, that might be different based off of the sponsor and the hospital. We've seen some situations in which some tests are covered by insurance companies if they're part of the normal clinical care. But that really, again, is dependent on the sponsor and the hospital that you're working with. And I'm going to just highlight a few of the main kind of risks, benefits, and outcomes for ASOs here. This is really simplistic and covers the basics, but ASOs are generally meant to slow progression and or reduce disease burden. They're not something that goes back and changes symptoms that have already occurred. They kind of slow the continuation of a condition going forward. For KCNQ2, this could mean the reduction of seizures or the opportunity for additional developmental milestones. However, we don't. This is an experimental. Clinical trials are experimental designs, and so we don't actually know for sure what the outcome might be. It's also important to know that in all clinical trials, there can be side effects of both the experimental therapeutic and the procedures that are part of a study. These can vary based off of the clinical trial design, and it's something to discuss with the team that's running any study that you're a part of. Aubrey's going to talk a little bit more about ASOs for KCNQ2 in her section, so we'll get into a little bit more detail then. And I will then pass it off to Emma.
Amazing. Thank you, Margaret. That was really great to get some background on all of this work and how we get to ASOs and what they do for KCNQ2. Thank you so much. All right, so I'm gonna change tracks a little bit and talk about long-read sequencing and how we can use it as a tool to help us design and target different sites in the genome for ASOs. So just to start, I wanted to give a really brief overview of different types of DNA sequencing. This can be an area that's really hard to navigate. There's tons of different sequencing out there. You'll hear terms like whole genome sequencing, whole exome sequencing. You can have panels and targeted sequencing and all different kinds of things. But there's sort of two big methods when we think about DNA sequencing, and that's short read and long read, which you see pictured here on the screen. And the focus of our work in this project is mostly on long read sequencing for a few reasons that I'll explain. So short read sequencing, as you can see, creates short chunks of genetic sequence, and then those chunks get merged together to create sort of a consensus sequence for the gene or for the whole genome, depending on what you're looking at. But one disadvantage of this method is that, as you can see here, this can create small gaps when those chunks are combined together, which can not be great if you're trying to understand a long stretch of DNA and the sequence there. So long read is really advantageous because it creates a really cohesive long stretch of DNA sequence that's not interrupted by gaps, and you can better assess sort of structural variants, just get a sense of genetic variation across a larger span of the genome. So again, that's what we are kind of focusing on here. For a bit of background on genetic variation as a whole, there's sort of two types of mutations and variants that we'll talk about today that I wanted to explain a little bit before we dive deeper. So the first is on the left, a pathogenic mutation. And these are mutations that cause disease. So for KCNQ2, every person who is diagnosed with KCNQ2, most people, have genetic testing done and they identify a pathogenic mutation in the KCNQ2 gene. And you can see an example here on the screen of what that type of mutation might look like. So C dot has a number, it has a base change, and that's describing, the change in the nucleotide sequence in the gene that creates a change that's in some way detrimental to the protein. Pathogenic mutations might be different in every single patient. So theoretically, every individual who has KCNQ2 could have a different pathogenic mutation. And this makes it really tricky to design a drug because if every patient has a different site that needs to be targeted, that could be really difficult to create something that is applicable to, a broad population, a rare disease population. So there's a little lightning bolt here on the screen because mutations, especially those that are not inherited from a parent, so de novo pathogenic mutations, sometimes we think of them like lightning strikes. So they're random, they can happen kind of anywhere in a gene, they are damaging to the protein in some way, and yeah, it's kind of this like the small little bolt that's changing the sequence in some capacity. In contrast to those pathogenic mutations, there's also natural benign variation in genetic code. So across the entire genome in all people, there is natural genetic variation. And most of the time, when we classify them as benign, these changes are silent and they're common. So happens everywhere. They're not impacting protein or causing disease, but there is a difference in genetic code between people or across different sites in the genome. So for example, 30% of us who are on this webinar right now might share a a change in the genetic code at one specific site in the genome. And that number can range anywhere from, 0.0001% up to 100% of people. So the reason that we mention this is because we can use these natural genetic variations as a handle, which is why there's a picture of a handle on the screen, as a way to grab onto the mutant copy of a gene and help us identify target sites for an ASO, which I'll describe in the next few slides. Okay, so expanding on this, how do we get a handle on the mutant copy? So you see here two different copies of a gene, say KCNQ2. And as Margaret mentioned, everyone inherits two copies of a gene. You get one from mom, one from dad. And in the case of when a pathogenic mutation is present, you have a mutant copy of the RNA, which you see at the bottom. We've noted a theoretical dominant negative mutation here in red. And then there's a wild type copy of the gene. And wild type just means the copy that doesn't have a mutation. So essentially the reference normal sequence. And as Margaret explained earlier, we want to target the copy of KCNQ2 that has this pathogenic mutation, and we want to knock that down, right? So if in this case, if this mutation is detrimental to the protein, we want to reduce the amount that is expressed. And one way that we can do this with ASOs is by targeting the mutation directly. So as you can see here on the screen, the little blue squiggles, we could design ASOs that bind directly to that mutated sequence, where that mutation is, where that little lightning bolt is, and that can be a way that we can try to knock down that mutant copy of the gene. In addition to focusing directly on the pathogenic mutation, we can also make use of these benign natural variants as I described. So we can use these genetic variations as a handle and design ASOs to these benign variants to also work and knock down the mutant copy of the gene. So as you can see here, there's some yellow spots on the mutant copy RNA. These are not affecting, they're not leading to disease. They're just, again, naturally occurring variations, some small single nucleotide change in the genetic code, but because they're present and because they're on the same copy of RNA as the mutation, we can also design ASOs to those sites and similarly knock down that copy of RNA that we don't want overexpressing in the body. And finally, these sequence differences, there are also sequence differences present on the wild type copy, and these can also be utilized. So we can also identify through sequencing changes that are on the other copy of the gene, and then design ASOs in that space as well to redesign against the pair of the wild type copy, and then we can knock down the mutant RNA as well. Okay, so in this situation, in this schematic you see here on the screen, we're visualizing ASOs that are treating a single patient. So we have patient 1, patient 2, and patient 3, and they all have a little blue squiggle ASO targeting their pathogenic mutation. So in this case, we have 3 drugs for 3 different patients. But ideally, we want to identify more common drug targets that could be applicable to several patients at once instead of just a single patient at a time. And so what is shown here is a situation in which we've identified some of these benign, naturally occurring genetic variations in the gene that are aligning across multiple patients. So on the left, you see a site that has 2 patients that both have the genetic change. And then on the right, you see there's 3 patients that all have a change in the genetic code in the same position of the gene. And so if we design ASOs to target these sites, we're actually able to treat multiple patients at once, given that they have these same genetic variations in common, even though their pathogenic mutation might be really rare and might be different from someone else. And this is really great. This allows us to increase kind of the capacity of ASOs and really expand what our options are for treating patients. And this is where long-read sequencing is especially helpful because it helps us determine which variants are linked to the others on the same mutant RNA copy. So we are working to identify these targets by sampling the population of patients that have KCNQ2. So we've been working to figure out what the landscape of variation is and what some of these sites might be. And we're thinking of this as sort of a portfolio strategy. So by making use of these common benign variants, we have predicted that a portfolio of 3 ASOs could apply to about half of the KCNQ2 DEE population. So with just a few targets, by making use of these common sites, we're able to reach a much larger group, a much larger portion of the population than if we were focusing on just an N of 1 approach with a single patient at a time. All right, so to give you an update on some of our current progress, this figure summarizes our pilot long-read sequencing study, so what we've been able to accomplish so far, and the data that we have from 6 different patients with KCNQ2. This group of patients is a representative selection of the KCNQ2 patient population. It's only 6 patients, but it helps provide insight into some of these shared common variants that might be present in the population. I know this figure is a little bit busy. It might be a little bit hard to understand, so I'll try to walk you through what each row and each column is showing us. So first of all, each row of dots that you're seeing represents a single patient. So M1510 is a patient, M3667 is a patient. Each row is a patient. And then each column, and shown in the bar graph at the top, counts how many variants are shared between the patients identified below. So on the left-hand side, we see a grouping of private variants that are unique to each patient. So for example, M3666, all the way on the left, is showing that they have 93 unique variants. And then the patient next to them, M1513, shows that they have 49 unique variants. So all the way on the left, we're just seeing a summary of what variants are just unique to that patient, right? So like not shared across the population. And then as we move to the right, we start to see variants that do show up in multiple patients at once. So all the way on the right are the variants that are the most shared across this small cohort. And we see up to 4 out of our 6 patients all the way on the right sharing these mutations. I don't know if you can see my cursor, but over here in this section, all 4 of these patients would share the same variant. So there are about 10 of these common variants that show up quite frequently and again are found in multiple patients. And these common variants are potentially interesting candidates for ASO targets because they could help multiple patients at once, which is our goal, right? So being able to identify some of these sites where we're seeing hits across multiple patients is exciting and something that we are excited to learn more about. And we have a few other analyses in progress in the same work to try to add a few more patients here as well, to just continue to understand what the landscape of variation is, how many variants are shared across individuals, these benign common variants, and of those variants, what could we possibly target with an ASO? So just to wrap up, I wanted to say that our pilot study was small and 6 patients as of now, but it did give us the confidence that ASOs could be designed that would help multiple individuals at once, which is definitely the goal. We do recognize that there's a need to do long read sequencing on as many patients as possible in order to best understand the whole KCNQ2 population. And so expanding this effort will require collaboration between research institutions, clinicians, and also advocacy groups like KCNQ2 Care and all of you that are working on this. So excited to see what could come in the future. And now I will turn it over to Aubrey to close us out with the last section.
Thanks, Emma. So with all of this background that Margaret and Emma provided about ASOs and long-read sequencing, I'll try and connect this all together and talk about how we're applying this information to KCNQ2 specifically. So our lab has been studying ASOs and KCNQ2 for the past 7 years. You may have heard about progress in this space, so Brooke invited us here today to give you an update on the status of our program. So I'll start by giving you a brief overview of how we design and screen ASOs, followed by an update on where things currently stand for this program. So we start by designing ASOs targeting variants across the gene. So like Emma said, we can design them against either the pathogenic or benign variations, and we will tile them across the sequences in the gene. And then members of this community have generously donated their cells to research, and we have some of them growing in our lab. And to determine if an ASO is having our desired effect, we treat patient neurons in a dish that we have derived from KCNQ2 stem cells, and we measure how effective the ASOs are in reducing the amount of KCNQ2 mutant RNA. Okay, so Margaret explained earlier how selective knockdown ASOs can target just the mutant copy of a gene and degrade the RNA while leaving the normal or wild type copy intact. So that's the goal of what we want a good ASO for KCNQ2 to do. We want to reduce the wild type copy as little as possible while knocking down the mutant. So this graph on the right shows RNA levels on the y-axis, which is the amount of each of the copies of RNA, with wild type in blue and mutant in red. And the horizontal or x-axis, we're looking at the measured levels of those copies of KCNQ2 when cells are treated with increasingly higher doses of an ASO. So as we move across the plot to the right, the cells got a higher and higher dose, and we're seeing how the levels of each copy of the gene change. So this plot shows an ASO with good selectivity because you can see that the blue bar for the wild type copy stays relatively level, even when we go to a really, really high dose, but the red bar decreases in a dose-dependent effect as we increase the amount of ASOs that the cells are treated with. In addition to testing ASOs for efficacy, they undergo a series of safety studies. So there's a few different ways that we can test if a drug is safe. They undergo rigorous safety studies that we design alongside toxicologists and get input on from the FDA. And this involves a combination of studies in human cells to look for inflammatory responses. We have a couple of different human cell lines that we test in, including donor blood to see if a human immune system has a response to the ASO being added into the cells. And we also look for off-target analysis in human cell lines to see if there are any unexpected impacts of introducing the ASO to a human cell and other parts of the genome to ensure that it's specific to KCNQ2. And then we do mouse studies. We inject the ASO directly into mouse brain to assess for signs of neurotoxicity. And then we do prolonged rat studies to help us define what the highest tolerated dose of an ASO is. So at the end of these toxicology studies, when we can see what the highest amount of ASO that a rat can tolerate is, we define that number and then we divide it by something like 30 to determine the highest possible dose that would ever be given to a human to make sure that we have a really big safety margin. So this is an overview of our screening progress to date for ASOs specific to KCNQ2. We've designed over 2,500 ASOs against 86 different target variants in KCNQ2, and we've ranked our designs and screened the top 1,300 of them in patient cells. We found that 86 of these met our thresholds for efficacy and selectivity in cell lines, and then we prioritized the best ones of those for a series of safety studies. And so far, 5 of them have been tolerated in preliminary cell, mouse, and rat safety studies. We're waiting for the final results from these safety studies, and then we will be able to nominate our top lead for a full, what's called GLP toxicology study in rats. So what comes next? A clinical lot of the ASO will be manufactured once we've selected one lead and used in a rat toxicology study. So this is a 4-month study where the animals will be closely monitored and receive repeat doses of the ASO. After that study is complete, the tissue from those rats' brains will be studied to assess that the drug has gotten into the right cells in the brain and that It hasn't caused any pathologic findings in the tissue. And this will all be assembled into a report. GLP stands for Good Laboratory Practices. It's like a set standard in the industry for the type of toxicology study that the FDA requires. So once we've completed that study, we will submit an IND, which stands for Investigational New Drug Application, to the FDA. And this application is a package that includes all of the data that has been generated for an ASO so far. So demonstrating the safety and efficacy of the lead along with the full toxicology report, details of the manufacturing process, release testing on the drug to ensure that it's safe and clean, and a clinical protocol that will have been written specific for the first patient in the trial, and would be amended for any additional patients. So this protocol will include personalized safety and outcome measures to monitor the patient throughout the trial. So these are the things that Margaret was talking about, like clinical assessments, EEGs, MRIs, all of that will be laid out ahead of time. And the FDA will review this package and all of its data closely before providing permission to proceed with patient dosing in a clinical trial. And in our ASO trials, our number one goal is to assess the safety of this strategy. As Margaret said, this is still a therapeutic hypothesis, and we're still performing experimental science with this therapeutic. We don't know how sensitive patients might be to knockdown of KCNQ2, which is why they'll be monitored closely, but ultimately we're trying to test if this hypothesis is safe. And then our second outcome measure that we'll be looking at is benefit. So outcome measures may take some time. Some ASO therapies for similar diseases have shown that it can take months to years to see improvement in some features of the disease, and some symptoms may not be impacted at all. So it takes time to observe the initial patients in one of these trials using close monitoring by families, clinicians, and through repeat assessments. So this is a hypothesis that we would be testing in a small, we call it N of 1 to N of few trial for the first patients. And then ultimately we will learn from them and figure out how to proceed forward after that alongside the FDA. But as Emma mentioned, we believe that having a portfolio of ASOs is necessary in order to benefit the majority of the KCNQ2 DEE patients long-term. But first we need to start with one. I guess we can open it up for questions.
Thank you guys so much. That was just really fantastic. I'm really excited to have sort of kicked off this conversation with the community. I do want to make sure they have lots of time to ask their questions. I have lots of questions as well. And there's two in the chat that I want to read for you in just a second. I just want to let everyone know that this will not be the end of this conversation. If there's questions we can't get to here today and they're in the chat, we'll write them down. We'll work to get some responses back out to the community. And also, sometime in the relatively near future, we're also going to be starting scientific updates that the community will get on a regular basis. And in there, you're going to see a form where you're going to be able to fill out questions that you have. And those questions are going to come to myself and Michelle, our scientific team here at KCNQ2 Cure Alliance. And so we can be answering questions on an ongoing basis. So just wanted to let you know, lots of opportunities to keep the conversation going. With that, I'll just read these two excellent questions from Tracy in the chat. As you move to clinical trials, how many years can be expected prior to the more general availability to the KCNQ2 population, and how might that impact efficacy in older children as they'll age, maybe while the clinical trial is happening? And then a second sort of related one, are there other ASO trials underway in similar neurological mutations, and how have they been going? Not sure who wants to tackle that. Let's see who comes off mute first.
I can
Aubrey's
start.
got it.
Yeah.
Great.
And then maybe Margaret can chime in if I miss anything. So as we move to clinical trials, how many years can it be expected to run prior to more general availability? And how might that impact efficacy and development in older children? That's a great question. It's not one that we have a very clear answer to right now. I think that we would continue to assess the initial patients until we and our oversight committee and the FDA believe that it looks like there is a significant safety margin and possibly signs of measurable outcome measures before it would be available to make it more generally available. That being said, some of these ASOs, like we said, they can be N of one or N of few. If there are other patients that are found that could be candidates for that ASO that have a severe clinical picture, regardless of age, there may be like a handful of patients that are eligible to be in the initial cohort. But yeah, to this point, we haven't had an ASO that targets a disease that's generally available to the population that has made that jump. But we do have a couple of our trials that are running at Boston Children's Hospital now, where we are starting to expand and recruit patients more broadly after, I think it's maybe been 3 to 5 years of treating the initial patients and monitoring them.
Yeah, I think that's a
Yeah.
sorry, it's a good point too, that while we've been talking about this platform approach, it's also very possible that the first ASOs to make it to the clinic would be focused very small, in the sense that they might only work for one or two people. And that's not necessarily by design, but more by what was most readily targetable with an ASO or some of the science that came through. So this whole idea of like a platform or kind of portfolio approach is something that we're working on that's somewhat new to this program. And so that's also something that we're kind of starting to think about and expand, but it's slightly different than some of the work that's been ongoing in the past. And for the other question, well, also in general, there have been several ASO trials across the board. ASOs are a well-known therapeutic modality or type of drug. There are ASOs that can treat hundreds of patients and are considered kind of more follow the more traditional clinical path. And then there's been quite an increased number in the last few years of these N of one ASOs, some of which are also for epileptic encephalopathies and have shown to have varying levels of success. These are very new, and so it's hard to understand kind of the data from across the board in other institutions. But we're keeping our eyes out for more information to come out and come our way.
Thank you. Yeah, and again, feel free to put follow-up questions in the chat. And again, if we don't get to it in the course of this discussion, we'll follow up. Then I'll just move down the chat. Are there any confirmed efficacy studies in mice with KCNQ2 mutations, or is that something in progress? Is it a necessary piece maybe of the drug development program?
Yeah, that's a great question. There aren't yet, but there are some in progress. It is difficult because there aren't a ton of perfect mouse models available to screen these ASOs in. So in order to be able to test the ASO in a mouse, we need to have a mouse that has a human copy of KCNQ2 that contains the variant that the ASO is targeting. So there are a couple of these models that are in development that we're planning to start screening ASOs in. And those are ongoing, and hopefully within the next few months, we'll start to get some results back and see.
Fantastic. Yes, and I believe that is part of the Cure Epilepsy Award that you and the Jackson Lab will be collaborating on. So congratulations on that. Excited to hear how that proceeds. Kara has a question in the chat. Would you assume all ASOs would have the same clinical results? Would you anticipate one ASO may be more robust than others, depending on the target that it's targeting?
That's a good question, Kara. We don't know for sure. We would expect that the clinical results of an ASO would be dependent on the profile of its knockdown. So like that graph that I showed, we think that the best ASOs will be the ones that knock down the wild type allele the least, and probably the ones that knock down the mutant allele the most. We don't know how sensitive KCNQ2 is. It might only take a little bit of mutant knockdown to see a response. And dosing in the clinical trial is something that can be adjusted by the clinician over time. So they can kind of be monitoring. And like Margaret said, since these ASOs wash out after a couple months, depending on how the patient reacts to a dose, you can kind of increase or decrease for future doses as you go.
It's also important to remember that we're going to be looking at very small population groups, like people who have actually received the drug, and everybody looks and acts and is different. And so what's measurable in one person, or what differences you can see after an ASO in one person, might look different in another person. And it's really hard to tell if that's because of the ASO itself or if it's because we're all different. And so some of those like subtleties and nuances are actually really hard to piece apart, and we've seen it across some of our other clinical trials where we have multiple participants, where it's quite hard to tell what's the result of a drug, and what's just kind of different for each of us, and how different people kind of present with their symptoms as a result of KCNQ2 changes. So there's like layers on layers for that question for sure.
Yeah, thanks for that. And I guess, so for those not familiar with my background, I work in genetic medicine as well. And I've worked with some, viral mediated therapies where it's sort of a one and done treatment. But I suppose one of the benefits of ASOs is if if you try a particular ASO and get a certain therapeutic benefit and think there might be room for improvement, and a new ASO comes out, there is still opportunity to try new ASOs. It's not like you have to commit to one and it's the one and only. So I think that's what's sort of exciting about ASOs. It's a ever evolving field, and I think we can only get better and better at it, which is exciting. And so Brian has a number of questions, which I greatly appreciate. I'm actually going to drop down to his third one first, because I think we already kind of touched on it a little bit, and I wanted to bring the n-Lorem piece into the conversation, because that's something that the community is a little bit familiar with. Obviously, some of our families have participated in the n-Lorem program. For those of you not familiar with n-Lorem, and if you want to know more about it, again, please feel free to reach out to any of us at the Cure Alliance. So n-Lorem is working on ASO approaches, I think, that are not dissimilar to what you're describing here. They've obviously moved into IND approval for other indications, and maybe you can kind of just talk about, why, maybe part of the question, or in my mind, is why do we have to go through the same process over and over? And maybe that's as simple as it's technically a different drug, right? And does n-Lorem and their process sort of predict anything about how your development will kind of move forward?
That's a really good question. I guess it's important to start with that we're like a distinctly different group from n-Lorem, and we don't have open communication with them constantly. So we aren't completely up to date on what the progress of their KCNQ2 program is. We can't speak for them, but in general, for other ASO programs, like Emma mentioned, we think that in order to treat the majority of the KCNQ2 population, we will need a portfolio of ASOs. So having multiple people working on getting these to the clinic is ultimately a good thing for the community. And the FDA just a few months ago put out a new pathway called the plausible mechanisms pathway, which is a way that we can get drugs for rare diseases through approval, like regulatory processes, more quickly and efficiently through like umbrella programs. So I think that there is the possibility to kind of like learn from what n-Lorem is doing and work together with them to kind of build a collective data package for ASOs for KCNQ2 that can be used to inform different trials.
Great, thanks. And then following Brian's questions, I think this next one's interesting from like a neuroscience perspective. And you touched on the mechanisms of KCNQ2 mutations and these dominant negative ones really being this sort of problematic protein getting in the way of the functional copy doing its job. And so the approach is to sort of silence or downregulate that copy, that's the problematic copy. And so do we know the dynamics of KV7 channels and how quickly kind of the system can recover, so to speak, once that dominant negative copy is no longer being expressed?
I don't know that we know for sure, but we do know the half-life of an ASO. So we kind of flood the system with enough ASO that should stay around for about 3 months. So as turnover is happening, there should be enough ASO available to then like bind onto the next mutant copy for that duration of time. And then there's kind of like a washout period after that.
Yeah, and that's something that's often sort of defined in the clinical trials as well, right? How frequently you might have to dose to kind of maintain efficacy. And then Brian's last question was about the shared variance. And I don't know if you want to just swing back to that data slide where I guess maybe the numbers like there's the private variance, right? So some individuals just have a lot of variation, I suppose, in their DNA. Is that what we're kind of meant to take away there? And then it's really those towards the right-hand side where like the common variance, like the more and more we can get of that means the more coverage we're getting on a particular ASO. Is that accurate interpretation?
Yep, you got it. Yeah, absolutely. So just trying to kind of enrich how much of this right side of the plot we're able to get. And as theoretically more patients get added to this, there's more groups and more intersections and more variants we're able to pick up that might be possible targets. So yeah, theoretically over here, there's 5 + 1 + 1, so 7 variants that are shared amongst 4 of these 6 patients. So theoretically looking at one of those 7 could be something that's exciting and the most applicable to the most people.
Fantastic. And actually, while we're here, I might just interject with one question, although Tracy has another couple of great questions that we could maybe squeeze in. How do we expand on this? What can we do as a community? What are next steps? Do we get you more individuals to long read sequence? Like how can we support the work here?
Yeah,
Yeah,
it's a fantastic. Go ahead,
it is a
Margaret.
fantastic question. Yeah. I think definitely for now, I think we're focusing on kind of wrapping up this pilot study and figuring out logistically what do we need next to bring this to more people. Long read sequencing is something that is difficult to get from a clinical perspective. It's not something that's readily available. Like if you went to your geneticist or your neurologist and said, I'd like to have long read sequencing, it's not something that is like very easily accessible to all clinicians at this time. So it kind of has to be done during a research study. So as we think about expanding to a much larger group, I think we have to look at ways that we can make kind of a more efficient research study to bring in more people, to get that information, to get samples collected, and all of the data kind of aggregated in one place. Because I think at the moment there's kind of all these little pockets. So I think that's kind of the next step that we're thinking about is how do we expand this in a way that's sustainable and will help the KCNQ2 community grow over time and get this information into the hands of the people who might benefit from it, as well as to inform our research. So yeah, definitely something that we're thinking about. I don't think we've come up with like, the next exact step, but we're really excited about the prospect of expanding this work.
Fantastic. Yeah. So everyone stay tuned and know now that when I mention Tim Yu's group, Boston Children's, Aubrey, Margaret, or Emma, let's all show up and see what we can do to move this forward. So that's great. Appreciate it. We've got 2 minutes. Let's see. So I'll read Tracy's 2 questions and let's see if we can get them both answered. ASO treatment, maybe just kind of talking about ASO treatment versus gene editing. And if you want to kind of talk about pros and cons, where they're both at, and then like the BF&E or DEE defined, and how would someone kind of know that difference? How is it
Yeah.
maybe, yeah.
These are great questions. So for the first one, I think I mentioned earlier in the slides that ASO treatments are only going to go so far. The kind of furthest we can get is probably about 50% function of KCNQ2, which is still not necessarily without symptoms. It's related with this benign familial neonatal syndrome. And we're not quite sure what it would look like to take somebody who has the more severe presentation and change their genetic background. Gene editing is kind of what we think would be one of the options to get up to 100% KCNQ2 channel function. But that remains a challenge for conditions that impact the brain. The science as a field is still trying to figure out how best to get those gene editors to the brain safely, and how to manage additional symptoms that come with those editing strategies. So that is something that's ongoing. Again, one of the nice things about an ASO is that they don't preclude you from participating in other studies or other clinical trials or other kind of genomic therapies later on, because they do wash out. And so I think gene editing or gene replacement is something that we'll be keeping our eyes on for sure, as something that could potentially be further down the road, but I won't say necessarily more impactful, but would potentially give you the opportunity to get to 100% KCNQ2 function.
One thing,
Yeah.
one thing I'll just quickly tag to the end of that, which we did already touch on, but it's worth noting, I think, that gene editing is a permanent one-shot thing. So it is a direct edit to your genetic code. So that is something that lasts for a lifetime. And as we mentioned before, ASOs are not that way, right? So ASOs are repeat dosing, modular, so can be kind of dose changed over time, which has its pros and cons. Some people love that it's modular and flexible, and some people think that that's a pain to have to come into the hospital every few months. So yeah, that's just another thing to add about like practically the route of administration is very different.
Great. Is there a quick answer on BFNE versus DEE? Is it mutation based or
some variants or some mutations have been characterized based off of their channel function or how much of the RNA is present, but that hasn't happened for all variants. And I think it's a question that we're still kind of trying to navigate ourselves. Some of it's symptom based, or if it's been inherited from a parent, you can kind of start to use some of these puzzle pieces to try and sort out the variants. But I don't think our team at least has a super clear answer for that.
Yeah, I think to determine which patients would be candidates for a trial and had DE, we would probably work with someone like Sarah, who does the functional studies of the patient's cells to look at the impact of variants, and also work with your neurologist, because they have the best view of the clinical picture.
Awesome. Well, thank you so much, and for the 2 extra minutes as well. And thanks everyone for being here today and having this discussion. I look forward to more and more about therapeutic development for KCNQ2. And with that, have a good evening, everyone, and we'll be in touch soon.
Thanks, everyone.
Thank you.